All right, so this is part two of the high yield review for theirs. Where was I? Okay, so let's talk about a little bit of micro. Please remember that pseudoappendicitis happens with um, enterocolitica, urinate, like it's the fucking plague thing, but it's called Y enterocolitica. Basically, it gives you bloody diarrhea, and one of the things it does, it makes the right side hurt, and uh, that's why it's called a pseudoappendicitis. And it happens in kids, associated with dog feces. Um, it can survive in cold temperature. It's gram negative. It's the pin shape thing, the safety pin shape. It's called bipolar, like something like this. Uh, what else? Yeah, don't confuse this with the plague. That's urinatus pestis. Really bad with names. Okay, hemolytic uremic syndrome. Basically, the sequence is these bacteria, which is either E. coli, enterohemorrhagic E. coli, or Shigella. Is it Shigella? Yeah. They produce a toxin that I need a bigger vessel. Demonstrate this. Produce a toxin that damages the vessel, right? What it damages specifically is it's kind of like an atom 13 or whatever protein. But long story short, that after this damage, uh, the platelets will become way more likely to bind to the vessel, right? They bind and they get activated. This is platelets that I'm going, and they kind of like activate other platelets, and you'll have this all over the place. So now this, when your red blood cells are moving along, they get kind of cut in half with like these platelet structures, right? So they make like these cells, red cells that look like they're cut in half, and these are called schistocytes, right? Schistocytes. So because so important distinctions here from DIC, for example. One of them is that if you notice, we are just activating platelets. We're not activating our um, like the components of coagulation system. So we won't have PT and PTT will be normal, right? But bleeding time will be high. Uh, these will be normal. Bleeding time will be high, and then the other thing that's going to be high, the other thing that's going to be low, is related to bleeding time, is that our platelets is going to be low. And so we are over consuming our platelets. We are forming clots where we shouldn't, and we're not forming clots where we should. So we're also going to get bleeding out of like places. Uh, and this usually happens in kids. Oh, and one more important thing is that this can happen. A lot of this stuff can happen in the kidney, and kind of decreases kidney GFR, decreases kidney's functionality, and as a result of that, we get the hemolytic uremic part, right? So the hemolysis is here for hemolytic, right? But the uremic part happens when all this happens in the kidney functionality goes down, so you get a rise in your creatinine level, right? Okay, H. pylori. There's a bunch of uh, what's it called? Viral factors you should know them, but I want to relate it to the path is that it's the most common cause of duodenal ulcers. 95% of duodenal ulcers as a result of H. pylori. Something about 75% of ulcers of the stomach are as a result of H. pylori. But it's important to know that when you have it happens for different causes. In the stomach, ulcers from H. pylori happens because of virulence factors of helicobacterium, right? Which is like this bug. But in the duodenum, it happens because we have as a result of the bacteria we have lower levels of somatostatin, and so it loses the protective function of the mucus. Either way, what's important to know here is that the gastric type of ulcer has a chance of turning into gastric carcinoma, and when it does that, it changes into the intestinal type of gastric carcinoma, right? That's not the one that's associated with Singet cells. It's uh, that one is diffuse type. Uh, C. diff. How do we diagnose this? We look for toxins uh, in the we look for toxins in the basically in the poop. We look for the alpha or beta toxin. Okay. Um, it's not. I think I'm not remembering. I may not be remembering properly, but it's not useful to do PCR because like a lot of people might have C. diff, but not necessarily the toxic type. So we look for the toxins in the poop, the feces, the liquid feces. All right. So let's leave dextra dexamethasone suppression test for last because it wants a lot of explanation. It's chewing on a antacid. Okay, let's talk about maternal diabetes and neonatal respiratory distress syndrome. Basically, what you got to do, maternal diabetes, or what it means is that it has the mom. It's usually type 2, right? Um, so that means there is high insulin in the mom, and insulin, what it does is that it decreases something really important, and that important thing is the like this stuff. What is it? It has a nice name. The surfactant. So it decreases surfactant in the baby's lungs. So high insulin leads to decreased surfactant uh, in the baby's lung, right? So that's why there's an association with maternal diabetes and NRDS. And what do you do for this kid? You give him corticosteroids before the babies do, and that kind of matures the surfactant production. Remember that surfactant is produced by the type 2 pneumocytes, and it's stored in lamellar bodies. Random point upcoming next is that diverticulitis, the risk factor for it is fiber. Remember that the first reason we even have diverticula is because the large intestine that looks like this. The reason it looks like this is because like parts of it like keeps contracting, right? And so when you have low fiber, feces tend to be harder. That means when it's contracting, the pressure builds up higher, and you have a higher chance of a weak point here and like kind of like ballooning out over like 70, 60, 50 years. You get like these weak points, so you develop more diverticula, and when you develop more diverticula, there's a chance of inflammation that leads to diverticulitis. Remember, diverticulitis happens more in the sigmoid column, and with the explanation I gave you, you should be able to tell me why. I hope you paused and tried, but the reason for that is as the water is getting reabsorbed in the large intestine. I mean, one of the reasons for that is so the water is getting reabsorbed, and by sigmoid column is where it's like the feces has is the driest, is the hardest, so you'll get like the highest pressure buildups, and you'll get higher chances for these kind of like these things happening. Um, okay, endometriosis. So I'm just gonna highlight this so I don't remember, I don't forget it. Actually, let's do it now. Dexamethasone suppression test. So basically, let's write our scenarios. We got syndrome of inappropriate, not inappropriate ADH. We got let's say syndrome of inappropriate ACTH. That's not an actual thing. I just made it up, but it makes sense, right? We got pituitary ADH, ACTH, right? And so in this case, it's a lung cancer ectopic. ectopic, probably lung cancer, small cell at that, producing it. In this case, it's like adenoma of pituitary, and in this case, you just have cortisol. In this case, it's an adenoma of what? Of the adrenal. All right. So, and let's add a fourth case that's just like nothing's going on. That's sort of like negative, right? And let's draw a bunch of lines to make things look cleaner. All right, let's learn this once for all if you haven't. All right, so we have our low dose here, we have our high dose here. So when you give someone the low dose, um, there is normal person, and what is this? These are measure of cortisol. All of these places are measure of cortisol. What I'm writing is measure of cortisol. Understand that, please. Right? You're measuring to see if the person's cortisol is responding to what we're doing. Okay, so let's start with the most. Let me minimize myself. Move myself here. All right. So with a high dose, a normal person will definitely downregulate their cortisol a lot, right? They even do it with a low dose, and that's what differentiates it with a normal person with any type of pathology, right? So with syndrome of inappropriate ADH, even a high dose doesn't do anything for us because these cells are crazy, crazy cancer cells that are neuroendocrine. They're not even supposed to be doing this in the same place. So, of course, they don't have negative feedback. They don't care. They're just producing stuff. But these cells, these are adenoma, these are kind of like nicer. These guys are, um, so these guys will respond to a high dose. So, high dose will lead to a decrease, right? And also, and for adrenal adenomas, they don't respond either because they don't have negative feedback at that level. Remember, the negative feedback that's placed is at a level of at a pituitary level. So with these measurements, we can easily distinguish this guy from this guy from this guy. But if you notice, this and this have the same type of presentation, right? So the thing about uh, uh, the, the adrenal adenocarcinoma is that your ACTH is going to be super low, right? Why? Because your pituitary is yelling at your adrenal glands to stop producing stuff because of the super high levels. They're just not listening. Whereas in this case, your ACTH will be super high, right? So that's the third column of differentiation is ACTH. Um, we'll differentiate these two. All right, let's talk about endometriosis. The basic idea with endometriosis is that endometrial epithelial tissue is somewhere it's not supposed to be. What are the common places? Myometrium is one, Uh, I think that's called adenomyosis. Uh, the ovaries is one. It can even go into the serosa of the intestines. It can go anywhere, right? But all of it has a bunch of things in common. First of all, it like creates brown stuff, and the most common thing you can think of is like the brown cyst, the chocolate cyst, and that's basically because every month the epithelium like really gets released. There's some bleeding, and over time that bleeding accumulates and produces some brown stuff. But it can happen as a chocolate cyst. It can be in other places. It will look like some sort of a description of something brown, something chocolatey somewhere. And the other thing is that these people, when they have their period, then they have extra pain, they have extra discomfort, stuff like that. So both of those things is pointing to endometriosis. We got this I've said a billion times right by now, but we got the diffuse type of stomach cancer, which is associated with lentis plastica, which is associated with lack of E catenaries, right? We also have the singlet cells here. And you also have Kirkenberg or something like that, tumors that is basically metastasis to the ovaries bilaterally, and that happens through seeding. We got a fibroid and a fibroadenoma. A fibroid, okay, so these are, I want to talk about them at the same time, but they're very different. A fibroid, um, and actually a fibroma. So fibroma is for the ovary, right? It can lead to Mage syndrome, which is basically you have the fibroid and you have some ascites and you have pulmonary, what's it called? Pulmonary, not edema. You have basically liquid in your pleura, right? Pleuritis. Um, I don't know if it's ritis or just liquid. So basically, you have this fibrous tumor in your ovary, and you have ascites, and you have some fluid. Pleural effusion, right? This triad is known as Mage triad. Um, so that's for a fibroma. A fibroid is the same thing as a leiomyoma, which is growth of the smooth muscle of the myometrium. It's associated with bleeding a lot during your every time you go through menses, and associated with decreased kind of like fertility, right? Fibroadenoma now is the fibro and adenoma. You know, it's two things. Is a benign growth of the breast. And last thing I wanna, and it's non-malignant and happens in like reproductive age people. But the last thing I want to touch on is that these are all. I'm not sure about a fibroma, the ovary mass, but fibroids and fibroadenoma, they're both dependent on estrogen, and with after menopause, you will when you lose estrogen, or let's say for whatever reason estrogen goes down, these masses will shrink. Gallbladder cancer is associated with gallstones. Why? Gallstones are associated with um, inflammation of the gallbladder. Inflammation of gallbladder leads to cancer, basically. Now there is this concept known as the porcelain gallbladder. It's kind of like you've had inflammation and like things in knowing and like using your gallbladder, that it starts getting calcified, and you can see it in X-ray or in imaging as like this calcified mass, kind of looks like an egg, and that's like super precancerous. You got to remove that. It can't be in your body because it's super high risk for developing into cancer. We got last. I don't want to talk about this yet because I'm not. Well, actually, I'll talk about it, but then I don't have any more concepts. I need to go like do more research and see what's going to be what I think is important to add for the next part. But before I finish, I want to talk about hemochromatosis. It has three things. Three things. Actually, let's talk about Wilson while we're at it. 33.2. So hemochromatosis basically, okay. So the first thing that goes wrong is that you have a mutation, right? This is for the hereditary form. You have a mutation that upregulates the reabsorption of the absorption of iron from your intestine, right? So you get more iron everywhere. You get it out of your intestine way more than you need to. So one of the places you get it, and iron goes through the Fenton reaction, which produces free radicals. So it damages everywhere that it gets stored. It damages beta cells. You develop diabetes. It goes into the skin. It gets a bronze color, and that's what it's called, bronze diabetes. So like person that's like a little bit tan as diabetes should already make you think of this disorder. You get restrictive heart problems. It can also lead to dilated heart failure. Why? Because you have iron there, and it's producing a lot of like free radicals, and you get cirrhosis, and that's obvious why, because a lot of it is stored in the liver, and it can damage liver because of the free radicals produced, and also these people a lot of time they develop what do they develop? They develop hepatocellular carcinoma, right? Same story with Wilson. The difference with Wilson is that what's wrong, like the gene that has a problem with, it's called ATP I think 7 beta. And here, this gene, uh, so the problem isn't absorbing too much. The problem isn't that we can't load it onto ceruloplasmin. So and it's not loaded onto ceruloplasmin. It's free, and it's kind of like going around and producing free radicals through the Fenton reaction. So in this disease, our ceruloplasmin goes down because uh, when it's not binded to uh, our copper, it gets degraded. Uh, yeah, we get cirrhosis with Wilson. We get super important thing. I don't know if it will be important for these exams, but the other name for Wilson is hepatolenticular degeneration, something like that. The lenticular part is talking about the lenticular nuclei of the brain, which means this disease. And if you remember, the lenticular part of the brain is uh, is basically the basal ganglia. So it has a Parkinsonian presentation. So, and it usually the other difference is as hemochromatosis happens in old people, Wilson happens in kind of young people. But so you get like those neurological issues as well as psychosis, stuff like that in Wilson. And classically, a more like younger person, hemochromatosis, you won't see like symptoms until you're like, you have around like, I'm not sure, I think it's 20 grams of iron in your body, and that happens after your 40s, something like that. Okay, last concept I want to talk about is ductal ectasia. Basically, I just want to say it's associated with a dilation of the nipple, and it happens in postmenopausal women who've had multiple children, and yeah, and it's associated with like kind of like green gray discharge. All right, until next time.